Hello everyone, welcome to this lecture 2. So in lecture 1, you have covered what are data. In lecture 2, we are looking at how do you get this data. So what we are talking about is your research strategies. Alright, so these are the contents that we'll be covering today. We are looking at how do you get the data, what are some of the sampling techniques. The main thing is so that we don't get biased. The second part of the lecture will be on the type of studies. Is it observational? Is it experimental studies, and what are the differences? The third will be looking at how do we summarize all these data in a tabular form. And the table that we're using today is your contingency table. Okay, so what are data? Data is essentially numbers. It can be numbers, it can be facts. There are two major types of data collection. One is called the primary data collection, another one is the secondary data collection. So let's say for example, you are in the lab, when you do any form of experiments, okay, it can be measurements even. Let's say if I want to collect the height and weight of all the students, that is observation. I can do experiments in the lab, or I can do survey. So how many of you are eating breakfast? So all these are called primary data. Secondary data works in a slightly different way. Secondary data is usually a compilation, which means that I can gather, sorry, my clock, I can gather data from primary data sources. Okay. Gather data from the primary sources and put into a secondary source. So I can have, for example, I will say that I'll send 5 of you to go and collect surveys from 5 different polytechnics. Then I collate them based on polytechnics. That become my secondary data compilation. Okay, now we come to actually how to collect data. Very commonly, we use surveys. Okay, so surveys, it can be through telephone surveys, can be you email questionnaires to people. Let's say, for example, you do your onset. That is essentially a questionnaire. Or you can go down to interviewing people. Okay. Now, when you do surveys, you have to take note of a few things. The most important part about surveys is the questions itself. Let's say if I ask, do you think that you are tall? The first question that I'll get is, what is tall? Tall to me can be different from tall to you. Maybe I'll decide, okay, anybody who is tall to me has to be above 185 centimeters. To you, anybody that is tall may be above 175 centimeters. So the question cannot be ambiguous. The same thing applies to your questionnaires and interview questions itself. Although in personal interview, you have the advantage of rephrasing your question or re-asking your questions, or even clarifying your doubts in multiple ways. So you can keep asking the same type of questions in different ways and try to solve it. But in questionnaires, you don't have the ability to do that. So crafting questionnaire is actually a very important skill. Thing about it is, in surveys itself, the ruler in survey is really your question. If the ruler can be interpreted by different people in different ways, it means that your ruler is technically flexible, and that is very hard to measure. Okay. So data can also be looking at other resources, let's say surveying records. How about records that we do not know? So I can, let's say if I want to collect data about Singapore population, I can go and survey previous year records, previous year birth statistics to look at all this. So they are records in the past. They may not be collected for the way that I want it to be collected, but I can look at that. Okay. Or I can look at direct observations. Direct observations, you have seen that before. For example, well, before you can go to, let's say, Tampines Mall, and you can see a person standing at the entrance clicking. So that is a direct observation of how many people are entering the malls or going out of the malls in a certain period of time. So that will tell you a lot about the traffic, how many people is visiting the malls. So those are direct observation. Okay. As a researcher, what we want is to have a sample to collect data from a large population. Okay. We cannot we cannot collect data from the entire population because it can be too large and it may not be feasible as well. So this is where we want to think about how are we going to get a suitable sample. Okay, next we want to look at samples and sampling technique. But before we go to that, we have to realize or we have to know what samples are. Essentially, a sample is part of a population. A sample must be representative of the population. So for example, if you want to, let's say, take your blood glucose, you are assuming that the drop of blood that you take from your fingertip represents all the blood that is in your body. It has the same amount of glucose, same concentration of glucose as in your body or in your whole circulatory system. That is what is representative. If your sample is not representative, then it will generate biasness. And biasness is what we want to avoid in statistics. Okay, so in this example, if you interview people at a street corner during the day, let's say you interview people at, let's say, 8 a.m. in the morning, then you are only interviewing the people that wakes up at 8 a.m. that is having to go to work or go to school. You're also, at that point in time, you're also looking at the people who are out at this moment. You are not interviewing those that, let's say, work in afternoon shifts or whatsoever. Another example is, let's say if I ask you to survey the hours of sleep in among TP students, and you stand at the bridge and you interview people at, let's say, 8 to 9 a.m. in the morning for 2 days. That itself becomes not representative. The data you collect is not representative because it represents only those people who come to school at that point in time. Okay, maybe they have lessons at 9 o'clock. How about those people or those students who don't have lessons at 9 o'clock but have lessons in the afternoon? So this becomes a big problem in statistics. You have to try to sample everything. Okay. A sample is essentially a small part of the whole population which you cannot study, but it must be representative. So in most research, we use some form of sampling strategy. In this case, we will actually explain to you the 4 common sampling strategies that you can use. They are simple sampling, simple random sampling, stratified sampling, cluster sampling, and systematic sampling. Okay, so for simple random sampling is the easiest method. You give each person or each subject a number. Then after that, you use a random number generator and you randomly generate a certain number and you sort based on that. Okay. It's essentially trying to draw lots. Let's say if I do a random sample of this class, I assign each one to you to a different number. So you count off from 1, 2, 3, 4, and so on, all the way to 20 or 50 students. Okay. Then I randomly pick these 50, let's say, numbers, and I pick randomly draw lots. Let's say I draw 5 of them. Okay, I do a shake in my box, and I just draw any 5 of them. That is essentially simple random sample. Okay. Stratified sampling is slightly different. We divide the population into strata. Within a strata, the subjects are homogeneous. So what does this 2-sentence means? So let's say if I want to sample this class, I can have different strata. I can strate you based on gender, males or females. Okay. If I strate you based on gender, within a strata, all of you are homogeneous. All of you are males and all of you are females. I can strate based on, let's say, ethnic groups. So I strate based on Chinese, Malay, and so on. So you are homogeneous inside. And within each strata, we actually do a random sample. So this can be quite important and quite useful. Let's say if you are trying to sample from stratas that is uneven. Okay, let's say from different income population. Okay, you have certain income and so on, or from different group, from different career types. So that is a different strata. Okay. The third sample we come is called cluster sample. Cluster sample is the opposite of strata sampling. Okay, first you divide the population into clusters, and then within it, the subjects are not homogeneous, and then you do a random sample. The easiest way I can do this, I can think about is, let's say I do a cluster sample based on your course. So automatically the clusters becomes your four or five different classes. Within each class, the subjects are not homogeneous. I sample 3 students from each class. Do a random sampling, I randomly pick 3 students from each class. So that in itself is a cluster sample. Systematic sampling, basically you order all the subjects and select every nth subject. Okay. So in this case, let's say a population is 1000, the sampling size is 40. So every 17 subject you sample. So how do we do this in real life? Very often we do this, let's say when we sample for quality control. Okay, let's say if you like ice cream, your others ice cream, okay, your others ice cream come in small little cups of 60 grams. So how do you know that it is really 60 grams? So maybe I sample every 100 cups that come from a certain machine. So I sample cup number 1, cup number 101, cup number 201, and so on and so forth. Okay, so that is called a systematic random sample. Multi-stage sampling is a mixture of all this. If let's say you have a very large population, you can have cluster sample and a systematic sample. So I can have 5 different ice cream manufacturing lanes, or 5 different ice cream manufacturing machines, and then I sample every 100th of them, every 100th cup. So 5 different machines, that is essentially a cluster, and I do a systematic random sample on top of that. Okay, so these are the common type of sampling techniques that we used. All right, once we are done with sampling, we will come to this stage of what are different types of studies. There are really 2 major types of studies that we want to look at. The first is observational study, the second is experimental study. Most of the work that you do in the lab, you're actually changing a variable and looking at the outcome. You change one variable at a time looking at outcome. So you did that in your primary, secondary school science before. Let's say you want to look at, for example, plant growth. Okay, you can change the amount of water, you can change the amount of, let's say, magnesium you give to the plant, you can change the amount of nitrogen you give to a plant, and look at the growth. That is called experimental study. You manipulate one or more variables. So let's say water, sunshine, and all these are different type of variables, and you see how these variables affect the end result. Those are experimental studies. But there are many areas in science that we cannot really do experiments in, and that will fall under observational studies. For example, we are looking at, let's say, disease progression. Okay, let's say how does the immune system react over the course of a flu episode, influenza episode. So that is observational studies. You really cannot do a actual experiment, but you look at the outcomes as you go along. So those are observational studies in itself. Okay. In essence, observational studies is only done or usually done when we cannot do experimental studies. Okay. So there is this concept of what a variable is. A variable is simply the item that you want to measure, or the item you want to change or you can change. Having said that, variables come from are split into two separate areas. One is called the independent variable, another one is called the dependent variable. The independent variable is the one that is changed or manipulated by the researcher, or sometimes known as explanatory variable. So let's go back to the plant study that we're looking at. Changing the amount of water, the amount of sunlight, the amount of water and the amount of sunlight, those are independent variables that you can change. What results from the independent or explanatory variable, which is the growth of the plant, that is called the dependent variable or the outcome variable. In simple maths, dependent variable is the subject of the equation. So let's say you have a straight line, y equals mx plus c, your regular straight line, y equals mx plus c. Your y is your dependent variable, or also known as the outcome variable, while your x is your independent variable, or also known as your explanatory variable. Okay, so let's come to observational studies. There are two different types of observational studies. One is known as cohort studies, another one is called case control studies. Cohort studies itself is separated into two different types. So you have a prospective cohort study and retrospective cohort study. Let's examine what are these terms prospective and retrospective means. Prospective is to look at outcome, which means that you are at this time, that means the current time, you are looking at what happens in the future. Retrospective is looking backwards. You are now at the current time, you are looking behind. Okay? So how does it look like? Prospective study will mean that, let's say if you have a certain cohort, okay, let's say you have a population that is influenza patients, they are now at time 0, the current time. You look at the outbreak progress of the disease outcome over a certain period of time. That is called prospective study. Okay. Retrospective study is you look backwards. So, for example, if you take, let's say, cardiovascular disease patients, okay, let's say a person has cardiovascular problems, you look backwards into the lifestyle, what may have caused them to have cardiovascular disease. So that's looking backwards in time. To put it simply, when you're actually reading a famous person, the life of a famous person, be it Bill Gates, Jack Ma, or whomever, okay, what you are trying to look at is what makes these people successful. So those are retrospective studies. When you're looking at a person's autobiography, those are really retrospective studies. Okay. Prospective studies will almost be taken. Okay, you know that Ivy League universities, let's say Harvard and so on, Harvard, Yale, they produce a lot of famous people. So prospective studies will say that, okay, let's look at current batch of 2020 graduates, see how they progress over the next 50 years of their life. So that is prospective studies. The term cohort itself, cohort is simply a group of subjects sharing a particular event. So a cohort study means following up over time. That is a term what longitudinal means. You are following up the patient. You are following up the subject. So back to our example of your prospective study, you are looking at the current batch of graduates from a certain university, see how they progress over the next 50 years of their life. Can you actually do cohort study with retrospective? Technically, it's possible, just that you're studying the person's history. Okay, the history can come from the person's journals, the person's interviews, and so on. Those are retrospective cohort studies. Okay, so here what we have is an example of prospective cohort study is the Rotterdam study. Essentially, they are trying to look at what are the factors affecting occurrence of different diseases. So they have cohort, they start off with a group, and they follow them up over a certain period of time. Essentially, if you do prospective study, it takes a long time. Okay, I remember the largest or the longest prospective studies is to look at what makes a person happy. So they actually follow groups of people throughout their lives. So essentially, prospective study can be very expensive as well. Okay. In this case, your Rotterdam study, they actually have people from different cohorts, and they actually look at them over their whole life to see how the lifestyle and so on affect different aspects of their health. So diagrammatically, this is what it means. You have the presence or absence of exposure. So in this case, you can be graduating from a University A versus not graduating from University A, and the investigator begins your research, and then you look at the outcome. So you're looking into the future. Okay. Now we look at retrospective cohort study. Retrospective cohort study simply means retrospect. You're looking at the backwards. So you have collected your data. So let's say you have collected 100 patients, or rather the case examples of 100 patients with, let's say, cardiovascular disease, or even, let's say, high blood pressure. Then what you want to do is to see what is the risk factor that causes high blood pressure. What is the risk factor that tells you the person is likely to have high blood pressure in the future? That is retrospective study. Okay, so the main thing is you can compare that. You're almost like taking yourself into the future. Okay, it's just a different time point. However, you have to take note about prospective and retrospective study is, let's say a person is consuming high fat diet. Does it mean that he will have, let's say, high cholesterol or high blood pressure or heart disease? So there is a certain level that you have to play with here. So sometimes we look at both prospective and retrospective study in a certain context at the same time. Okay, what do I mean by that? Let me try to explain it clearer. Let's say you know that from your retrospective study, you say that, okay, high fat diet or high sugar diet results in is a risk factor for diabetes. Okay, so you determine, let's say, high sugar diet is a risk factor for diabetes. Okay, then you have to look backwards and say that, okay, or you do a prospective study then, is does all the people having high sugar diet results in diabetes? You get what I mean here? Okay, so these two actually are two different sides of the same coin. Okay, you cannot say that high sugar diet will cause diabetes unless you do the prospective studies as well. Only when the prospective and retrospective study are aligned to each other, then you can have a cause and effect. Okay, so this is an important part. Okay. All right, diagrammatically for retrospective study, we are looking at already have an outcome. So from the outcome itself, we want to determine whether do you have the presence or absence of exposure. Let's say the outcome of diabetes. Do you have the exposure of high sugar diet or no high sugar diet? Okay, now we come to the second type of observational study, which is case control study. Now this is very similar to your cohort study with a few subtle differences. For case control studies, the two groups are already identified based on some form of variables. So the real difference is that the case control studies and cohort studies is that for case control studies, the outcome is identified, then you look backwards. Okay. For cohort study, the exposure is identified first. Okay. So it can be pretty confusing. Let's look at how we draw it in a diagram. Okay. Diagrammatically, a case control study will mean this. You actually have the outcomes already,
but you're looking backwards. So it almost means that you have the two groups of outcome, whether a person have diabetes or don't have diabetes, and you do a retrospective study on it. Okay, so this is pretty confusing, but it will become clear when you actually look at some of the examples later on. Okay, let's look at this example. I'll just give you a few seconds to read it. In 1967 in Great Britain, there's a concern whether occurrence of, let's say, blood clots happens in women with taking oral contraceptives or not. So to look at the outcome, or to look at the relationship, Vesey and Doll in 1969 studied the existing cases of this blood clot disease. Okay, so how do they study the existing cases? First, they identify women aged 16 to 40 that already have been discharged from one of the 19 hospitals and have had a diagnosis. So what type of study will best describe this? Okay. At this current point in time, at 1969, when the study was conducted, or 1967, number one is the outcome known? Yes, the outcome is known because these patients have already been discharged with a diagnosis, so the outcome is known. And then you're looking at a cause, a possible relationship, you're actually looking at the history. Do you have a control here? You do not have a control. You are not looking at the people who do not have this disease. So when you do not have that, there is no case control. The case control is out. You only left with retrospective cohort study. So this is a retrospective cohort study. All right. All right. After looking at observational studies, the next one we want to look at is experimental studies. So remember, experimental studies is where the variables, the independent variables can be changed by the experimenter. So this is very important. It's usually most of the experiments we do in the lab. Okay. However, what we want to tell you, or what we want to have you to understand here is how to actually design experiments, because any design errors can actually lead to a wrong conclusion. Number one is there must be randomization. Randomization simply means that there must be assignment to the treatment and control group or groups randomly. Okay. You cannot have a biasness in the treatment group or the control group. So the biasness must actually be distributed evenly. In this case, what we call as confounding variables. Later, we'll talk about confounding variables in the next slide. Okay. So indirectly, what you want is, if you are looking at the average, or rather you want to look at treatment A versus treatment B, chemical A versus chemical B for, let's say, cell growth. Okay, your cells is actually, the whole population of cells is deemed to be homogeneous. The cells cannot be pre-treated differently. It must be treated the same way. It must be mixed together before you go into the treatment and control groups. So in this case, there can be one or more treatment or control groups. You can have more than one control groups. You can have more than one treatment groups. Okay, so randomization is critical. After, another experimental design is case control. So case control, you are looking at yes versus no. Okay, one group having a treatment, the other group do not have a treatment but will receive a placebo instead. Okay, so what is a placebo? We'll talk about that in the next slide. The response variable is, at the end of a study, look at the responses in both groups. So placebo is actually very important here. Okay, so what is a placebo? A placebo is essentially what we call as a sham or fake treatment. For example, if I want to test whether it's a tablet or medication, let's say chemical A, useful to treat headaches. Okay, what I can do is I can manufacture one batch of real medicine or real chemical. The placebo is a fake chemical. Okay, it's just shaped the same way as a normal tablet. okay, as a treatment tablet, but it has no active ingredient inside, so it can be just starch or sugar. The main thing for medical research is placebo. There's actually this thing known as a placebo effect. Okay, it works this way. If you think that you are actually taking a real medication, psychologically and physiologically, you will start to feel better. It's the same thing. If you think that you are sick, you're actually feeling worse. So that is called a placebo effect. We want to actually deny this effect, and we actually want to cater for the differences. Blinding, we have double blinding and single blinding. This is more for medical studies. So double blinding works this way. You do not know, the researcher do not know who is in which group. So you actually receive all the data, but there's no labeling in the data sheet, who is in what group. Then you analyze the data. Okay. The patients themselves, the subjects, also do not know whether they are in treatment group or in control groups. Single blinded means that only one of the two, that means either a subject or the researcher, do not know. The other actually knows. Okay. So for example, let's put it in the exam context. Why during exam you should not, we ask for the exam script, you do not write your names on the exam script. It's because we want to say that the lecturer or the marker do not know who you are marking. Okay, that is single blinded, because as a student you know who is marking your exam scripts. Although you try to make it double blinded, in a way you do not know, the student do not know which lecturer is marking which questions. The lecturer do not know which student he or she is marking as well. So that's called double blinding. Matched pairs is very useful. We'll come to matched pairs more detail when we talk about statistical tests later. So what we are trying to do is using two matched individuals, okay, or the same individuals with having two treatment. More commonly is matched individuals, and this is where twins are very useful in medical studies, because twins have usually grow up in the same environment, they are genetically identical. Here we are talking about identical twins. Okay. One receive one treatment, one receive a control treatment, and they do not know which one receive a actual treatment, which one receive a control treatment, or receive different treatments. Their results are matched in that sense, or the same individual receiving two treatment. So let's say if I can ask you to test two different moisturizer. Moisturizer A on your right hand, moisturizer B on your left hand. At the end of 1 month, report the results for two moisturizer. So you say that you give a score, maybe 1 to 5 for moisturizer A and 1 to 5 moisturizer B. And you can say that the number 1 that you give for moisturizer A is the same as number 1 that you give for moisturizer B. So that is matched. But let's say if you have two person, one person I tell him to use moisturizer A, another person to use moisturizer B, then it's an unmatched pair, because persons A putting number 1 and persons B putting number 1, the two number 1 score of 1 is different. Okay? So matched pairs are actually very critical. Confounding variable by definition is that the influence of the variable is not well separated from the other variable. So yeah, you have actually two variables that is related to each other. Those are called confounding variables. Okay. For example, height is generally associated with weight as well. There is some relationship between height and weight. Okay. A taller person generally weighs heavier than a shorter person. Okay. Generally, not all the time. You have a very tall and very skinny person, or the other way around. But generally, there is a relationship. And you cannot tear these two apart. You cannot say that it's based on height alone or it's based on weight alone, because those are what we know as confounding variables. You cannot tear these two variables apart, and that's what makes it confounding. The next thing is what we want to look at is cause and effect. Does the change in the variable results in the effect? Okay. Observational study cannot, I repeat, cannot have a cause and effect. It may be correlated, but correlation does not equate to causation. This is very important. Correlation does not equate to causation. Okay. Only an experimental study can prove cause and effect between two variables. Okay. Why? Because in observational studies, there may be confounding variables, and there can be variables that is associated with something else. Okay. For example, we found that actually murder rates increase with ice cream sales, but we cannot say that people get murdered because they purchase ice cream, or we cannot say that ice cream purchase leads to murder. The main thing is it is just the number of people. Similarly, we found correlation between ice cream sales and the number of shark attacks. Okay. Why? Because when you have ice cream sales, it's usually in the summer months. In northern hemisphere, southern hemisphere, like in Singapore, it's the summer months. And summer is where people go to the beach as well. So the more you sell ice cream in the beach, the more shark attacks there is. Unless there's no relation between these two, it's just by the number of people. Unless you really think that eating ice cream makes you a bit sweeter and tastier for the shark. That's a totally different story. Right? So we can find a lot of cause and effect. In fact, we cannot find cause and effect from observation studies. Always remember this. Correlation does not equate to causation. Only in an experiment, then you can actually play around with the individual variables to see what is the result that it has on the dependent variable. You play around with the independent variable to see what's the result in the dependent variable. Okay. Okay, we have come so far. This is the last part of lecture 2, contingency tables. Contingency tables is to look at categorical variables. That means based on categories. So let's read this short paragraph. In 1986, there's a risk to find the risk factor between low infant weight, and one of the risk factor is the smoking status of the mother. So high infant weight, low infant weight, with mother smoking or mother non-smoking. It turns out that 59 out of 189 infants born with weight less than 2.5 kilograms. 74 of the mothers that smoked during pregnancy, and 30 of them has a infant weight born that's less than 2.5 kilogram. So as you can imagine, even for me reading it, it's actually pretty tedious. You have no idea. What we know is, can we put them into a table? So the column and the row is high infant weight, low infant weight, high infant weight, low infant weight, with smoking mothers, non-smoking mothers. Okay. So that is what we call as a contingency table. Okay, so this is how the contingency table looks like when we take the data and we put them in a 2x2 table. Of course, the table may not be just 2x2, can be n times n. Okay, it can be any rows, any columns. Generally, always remember, by convention, the rows represent the exposure status, treatment or so. So here we have two treatment status, smokers or non-smokers, smoking mothers or non-smoking mothers. Okay. The rows can have multiple. Okay. You can have treatment A, treatment B, treatment C, treatment D, and so on. Column in this case determines the outcome. In this case, will determine whether it's a low infant weight or high infant weight. Now, with this table, it makes our life so much easier. We can see that smokers have a total of 174 babies, out of which 30 are smaller babies and 44 are larger babies, and so on. So we know the comparison. This is what we want to do. It makes your life easier to analyze the data after that. As you can see, the very common contingency table is just a 2x2 contingency table. Factor, the exposure is yes or no. Disease, yes or no. So you have A, B, C, D. Total, you have A plus B plus C plus D. And from then on, we are able to analyze whether are there a presence or absence, or the main thing is how does the exposure of a condition or risk in the example in front, it is whether the mother is a smoker or non-smoker results in a potential change or weight of an infant. Okay, so we will actually be using these contingency tables quite often. Okay, and this is an easy way to look at it first. With that, we have come to the end of lecture 2. So thank you very much.